Our first speaker today is Dr. Kyle Wickings, and he is an associate professor in the Department of Entomology at Cornell Agritech in Geneva, New York. Kyle's research focuses on the ecology of soil arthropods and turfgrass insects. And the title of his presentation today is Tackling White Grubs in Your Lawn, Scouting, Decision-Making, and Sustainable Management. So Kyle, the floor is yours.
Great, thanks, Matt. And do you, again, have the audience view of my talk?
Yes, looks great.
Great, good to know. All right, well, hi, everybody. Matt, thanks a lot for having me, and it's good to be here. I hope you're all doing well. So as Matt said, I'm Kyle Wickings. I'm the turfgrass entomologist here at Cornell, and my lab focuses on soil biology. So a lot of the pests we deal with in turf are below ground. So that's why we're all soil ecologists here in this program. But we're going to talk specifically today about white grubs because they can be such a challenge in lawn settings. And we're going to focus a lot on using a very specific management strategy, using entomopathogenic nematodes or insect pathogenic nematodes for their control. So I just want to start by giving an overview of the annual white grubs. So there are 5 that we typically deal with here in New York State, and they're the ones you see pictured here from these great images from Dave Shetler. So we've got European chafer, the Asiatic garden beetle, Japanese beetle, northern masked chafer, and the oriental beetle. So these are the ones that we typically are dealing with here in New York and generally in the Northeast. So this is the life cycle. We'll talk about it a little bit better in more detail on the next slide, but this is the typical life cycle for all 5 of these insects with a little bit of wiggle room depending on the species. But so zooming in on this a little bit more, this is actually a great illustration right out of the IPM program, so appreciate using that from them. Right now we're right here in September at the beginning of it, and we've got white grubs. The eggs have hatched about a month ago or so, and they're maturing through 3 different life stages. So and all the while they're feeding on our turfgrass roots in the lawn, maturing towards what we call their final or third instar. So once our soil temperatures drop below about 50, they'll actually go deeper in the soil, and they'll go below what will eventually be the frost line in soil to protect themselves against freezing in winter. So they'll be inactive at that point, won't be feeding or anything. And they'll stay there until spring when soil temperatures return back above 50, and they come back up right into our roots and feed again for a pretty narrow window of time while the turf is greening back up. The roots are reestablishing the turf. They'll do that for a couple of weeks before they turn into a pupa. You can see that on both sides of this slide. If you look all the way to the left here, you can see that same stage. They'll pupate. That's basically the transitional stage from the grub into the adult form, which emerges from the soil back above ground to reproduce, and in some cases, like the Japanese beetle, to also feed as an adult before the process starts again, and the females burrow into the soil to lay eggs. So that's the cycle. Within this time frame, there are a couple different windows for intervening. And the first is typically what you think about with the window of grub control. And typically we're using something like a systemic insecticide here that has to be put out early around the time of peak adult flight. And the idea behind that is you need the insecticide present in the soil to be taken up by the plant in enough time so that by the time these eggs hatch, the material is present in the roots and can interact with the feeding insect. So that's why they have to go into the system early, because they're systemic and are being taken up by the roots. So some people think, oh, I'm hearing these grub applications are going out. This must be the window when I put out something like biocontrol nematodes. And that's actually a very different window, which we'll focus most of our time on today. But I just want to mention this because there's an important distinction there to make between this, the preventative approach, and what we're going to focus on, the curative application. So curative, when you hear that term and think about that window, it's really immediately when grubs are present. So you're not going to intervene until that population is present in the soil. So it's really confined around the stage of the young larvae spanning to the mature larvae being present in that system. And from here on out, this is what we'll be focusing on. So this involves a window of scouting, and that's good because that aligns with IPM, decision-making using data and the presence of an insect, its identification, and all those traits to have more informed decision-making. So a really good thing to be able to do, but it can be challenging to do. So we'll talk about how to do that and how to align it with the use of biocontrol. So you can use just about anything to scout, from a fancy sod cutter or golf course cup cutter to even just a spade shovel. We like to use those in our lab. Anything you can get a repeatable, uniform sample from the soil and turf with is totally fine here. The time of year for that is essentially starting a little bit earlier than now to right now on into early fall. So right about in the hot spot of when you can actually see and identify the grubs that are there. But it's a pretty flexible window, as you can see, keeping in mind that as these larvae mature, they become less and less susceptible to intervention. So on the other end of that, they're very, very hard to identify when they're really young. So you have the window shrinks a little bit. It's not super wide, but you do have some flexibility in there. So a lot of people think it's pretty daunting to go out and dissect an entire lawn, or say if you're a manager of a park or of an athletic field, it's a lot of space to cover. So our mantra is always start as broad as you have to and narrow it down where necessary. So in a typical quarter-acre lawn, we might take a dozen cores or so, trying to cover as much of that lawn as we possibly can. We make a little map in our notes, and everywhere we find a grub in our sampling, we record that spot. Say we found 2 in this example here. And then you have to decide, was it just a random grub that your shovel bumped into, or is it actually representing a hot spot in that area? So what we'll do then is we'll take some more intensive samples around that, the areas that we log grub presence at, to see if there are more grubs. And typically that will help reveal if there's a hot spot in the turfgrass or not. So you've taken what could have been a sea of grubs across this entire site and reduced it down to an effective area to actually treat if you determine it's necessary. So that's a really great way, if you can master this technique, to identify the areas that are vulnerable and need some kind of intervention, and also reduce the potential footprint of an insecticide if you decide you have to use one. That's just part of it, though. You found grubs. There's a few other steps in there, and a lot of that involves identifying who the grubs are, and we'll talk about why that is in a bit. But the process can be challenging. Knowing where to start, how to hold a grub, all of that can be tricky. So we put a website together, and I think Matt is going to share the link to this. If not, I can share it. The website itself is grubid.cornell.cals.edu. And the first bit of information is just some background. If you've never done this, we show the anatomy of the grub and exactly where you need to look to see the features that are important. And those features are the shape of the anal opening and this arrangement of hairs on the rump that we call a raster. So those two together are unique for each species of white grub. So it's a really good way to narrow down the species you've got. We list a couple other things, like you need to have a hand lens. Even a cheapo 15x hand lens will work for this, so you don't have to break the bank to get one of these. And a US penny, you'll see why that is in a bit. But we show how to hold the grub carefully to see the features you need to see, how to hold a hand lens, and all of that. So a really good reference page at the beginning to get you going. But the real launch page of the site, it looks like this. So you can, this is why I mentioned the penny, because we start to break everything down based on their silhouette size. And this just helps to start to filter the key down, because as you make decisions on the key, it starts deleting things that it couldn't possibly be based on your choices. So you just pick the silhouette that most closely represents the size of the grub you found. Say it's this one down here, you would click on it, and then it will take you to a series of questions, again, related to the shape of that anal opening and the arrangement of the raster, or those hairs. So you'll be guided through questions all the while as you choose options. It will be deleting species that it couldn't be based on your choices, until you get to a final species profile page, which has these really nice zoomable photos in here of the larvae, the raster and anal opening, and the adult. It repeats the threshold data, so you can compare the numbers per square foot or whatever to see if you need to make an intervention. We'll talk about that more in a bit. It shows you the actual life stage sizes for the 3 different instars of the larvae, a silhouette of the actual development for each, and when you would expect to see all the different life stages present in your lawn soil. And then at the end, we also give a link to the NicePad insecticide or pesticide portal, so you can look for products registered in New York State for the species you found. So a really great tool. It takes some mastering, but a really great tool to do that curative type of intervention and make an accurate decision. So you found the hotspot, now you know who it is, and you can then go decide whether or not you need to make an intervention. And doing so, there's a few different factors you need to take into account there. The visibility and the value of the turf. It's important, is it turf that you really don't care a whole lot about people seeing, or a turf that has a really high aesthetic value? It's very different in whether or not you would intervene with grubs. The history of damage at the site can be important sometimes. Obviously whether or not the pest is present and or susceptible. You want to target larvae, but as young as possible. But importantly, which species is it? So this is a threshold table here on the right, And you can see the numbers per square foot of soil vary a lot. So this is not just the annual grubs, but you can see across all of them, the numbers range from 3 individuals per square foot all the way, in one case, to 50 individuals per square foot. So being able to tell what species of white grub you have is really important there, because it can really help you decide if what you're seeing is likely to be problematic if you don't see damage yet, which is actually good, because you want to kind of hit that window before damage is present. Okay. So there's some trade-offs here. It's a little risky because you're intervening at sort of this late stage where it's the last chance you have before they go and disappear for the winter. Some of the conventional products that are present here can be a little higher environmental cost, but you're also scouting, you're narrowing down the footprint of where you might intervene to a small area. If it's a small hotspot, you can tailor your treatments. And it's importantly for our discussion today, this is where the opportunity for biological control actually enters the equation. So this is where nematodes come in. And Matt, I don't know if you want me to stop at all. If there are questions, you're welcome to interrupt me with any questions too, just as a heads up. I'm not really paying attention to the chat. So, so as I mentioned, this is the window when nematodes come into the equation, and you can consider their use. So let's talk a little bit about what they are and what their life cycle is. So this is a nematode at the top, carefully created in clay by a past graduate student of mine for this stop animation he did. But the nematode itself isn't the only thing you need. When you buy entomopathogenic nematodes, you're also buying a bacteria in its gut. And it's the bacteria that does all the killing. It produces the toxin that kills the insect, and it's what breaks the insect down and makes it digestible. And the nematode is really just the vehicle to move the bacteria throughout the soil. So these nematodes, they're not microscopic like bacteria, but they're quite small, and you would need a hand lens to see them. So it's very easy to overlook them in your soils. So these nematodes, when they're juveniles, are mobile. They'll swim through the soil pore spaces and water films, and they use chemical cues to find their hosts. So they find a white grub through chemical scent in the soil. Once they find it, they'll colonize the body through any kind of opening. It could be the spiracle, the mouth, the anus. Once inside, they'll sort of regurgitate or release the bacteria in their gut into the cavity of the insect. Those bacteria then produce a toxin to kill it. The bacteria start to feed on everything that they're breaking down and reproduce, and then the nematodes feed on the bacteria. So it's almost like they're farming their own food by delivering the bacteria to a food source. The nematodes, from all the energy they get from feeding on the bacteria, will reproduce, and this whole new generation of young nematodes will burst out of the cadaver and swim off to start the cycle again and find a new host. So there's a lot of interactions that have to happen in making that work, but that is the general life cycle and infection cycle for these nematodes. There's a lot of different products out there available, but it's really important to choose a nematode that fits the pest you're trying to manage. So there are nematodes that are really shallow and ambush their prey at the surface, and then there are others that are deeper and mobile in the soil. And these are the 3 names you'll see out there of nematodes that are well known for being effective against something like white grubs. So this top one is the most common. It's Heterorhabditis bacteriophora. You hear us call it H. bacteriophora or HB often, but a couple other species, Steinernema feltii and Steinernema scarabii, are also effective against white grubs. In some cases, not quite as much as this top one, but it's typically this top one you'll see labeled for white grubs. Okay. Formulations. So there's a lot of different ways you'll get these in if you purchase them on Amazon or from other kind of suppliers. They'll come in a whole different array of packaging. There's sponges they'll come packaged in, some alginate gels, water dispersed granules, vermiculite. The goal with all of these is to keep the nematode alive and viable for as long as possible. And that can be for months in some cases when left unopened. Once you open though, especially once you open and mix them with water to make an application, you really want to get them applied. So within a couple hours of mixing them with water, you want to make an application. There's a lot of instructions you'll get when you buy these because you're buying a live organism. They'll talk about refrigerating them, use by dates are listed all over the product, limiting exposure to UV because they're sensitive to that. But one important thing that we always do is when we buy them, we check their viability. We take a little bit of material, mix it with water, and let them sit for about an hour at room temperature. If they start to curl up and wiggle in the water, you know they're viable, and they'll just be kind of wavering in the water like that. If after an hour they all look like bananas or hangnails and there's nothing going on, no movement, chances are you have a package of dead nematodes. So very good to check, especially if you're going to go to the trouble of picking the right nematode, getting a good source and all that. You want to check and make sure what you bought is alive and it didn't get cooked on a loading dock. Okay, and lastly, application of nematodes. So a lot of people think this is overly complicated. You can use really complicated spray rigs for this sort of thing, all the way down to a simple watering can. There are just some things you need to keep in mind. You don't want to have screens or fine mesh that can plug up the flow of this material, because these are things that can get jammed into screens. So if you're going to have something like a spray nozzle, you want low mesh size or large mesh size, or completely pull the mesh out. You want to use low pressure. So it says less than 300 PSI. I think our sprayer in my lab is at 40 PSI. So it's quite low to begin with, but keep in mind that the pressure can damage the nematodes. We recommend about 1 billion nematodes per acre. That sounds nuts. It's like 20 million per 1,000 square feet. When you buy them, it'll tell you their numbers in similar units. So it's pretty easy to do the math for the area you want to apply on. You want to get the equivalent of about 1 billion per acre. Keeping in mind they're UV sensitive, putting them out at dawn or dusk, especially dusk, because you have the rest of the night for the nematodes to settle in without getting hit by sun. Putting them out when it's overcast is also really good, especially if it's going to rain, because that can help to water them in. If you have the luxury of having irrigation, you can also water them in with that. And you're really just trying to water them off the foliage and into the root zone, where they will settle and look for their hosts. So that's it. That's all I've got. And I don't know where we're at in terms of timing, but I appreciate everybody's time. If you have questions, we can either ask them here or in transition, or I'm also easy to find offline if you have more questions that you want to ask to me. So thank you.
Great. Thank you, Kyle. I appreciate your approach to this, providing sort of from beginning to end how to use these products to address the turf grass problems, especially the white grubs. We did have a question come in that I think is worth asking. So it was more of a comment that the person said, I do kill Japanese beetles if I see them, so we'll do the same to other beetles unless they are good for some other ecological purpose. So if these critters look like the typical scarab beetle that is a problem on roses or other things, would you advise killing the adult forms of these beetles, or do you think that there's some need to really know what we're looking at before we kill something?
Yeah, I think the latter is pretty important. A lot of these are maybe doing nothing except for going after nectar resources in the system, which also is not a bad thing. May not be giving you a huge ecological benefit, but doing no harm. So I think it is wise to learn to identify and recognize some of the common beetles. There's a lot of beetles there that you won't even know are there, even down at the soil surface. Really important predators that forage in the soil. And some that are very visible that are also giving you a benefit, including some of the wasps that are in the environment there as well. So definitely pays to learn how to identify the critters in your landscape. So that's a great question.
Excellent. Thank you. Another question that came in as Jody is loading her slides are the nematodes native?
That's a great question, Elizabeth. So these species are native, and you can find all of these species present in our local soils, even in the northern parts of New York State. What we tend to not see is densities or numbers of these nematodes high enough to control the pest on their own. But these are all native species that occur in our state and in our region. So really good question. And there's even some work going on trying to actually take the native strains that are actually in our soils and bump their numbers up. So good question.
Great. Thanks, Kyle. We're going to transition now to our IPM Minute. And this takes a quick look at a timely topic and provides some action items that hopefully you can take away for your weekend. And for this segment, I'd like to introduce Dr. Jody Green. And Jody is an extension educator at the University of Nebraska in Lincoln. And she's a board certified entomologist specializing in urban and industrial entomology. Jody is the co-host of a really entertaining podcast, the Arthropod Show, that you can check out. I'll put the link in the chat. And she has a really fascinating Twitter account that covers a whole bunch of different pests that you may experience in and around your home. And so today, Jody will be talking about pest flies that might affect you as we head into fall. So take it away, Jody.
Yeah, well, hopefully everyone can hear me. Thank you for letting me do this IPM Minute. Short presentations are always the hardest, I have to say. So let's get into it here. Let me see. So As Matt said, I am dealing with flies in the home environment. And identification matters because we need to find out where these pests are breeding so we can solve the problem long term. And so I'm going to introduce you to this, I'm going to call it the trashy trio. So we've got the common housefly. It's got a dark gray thorax with 4 black stripes. We've got the blowfly, which can come in green variety, bronze, or blue. We call them blue bottle flies or green blowflies. They're associated with meat and dead animals. And then we've got the flesh fly, which is more than 2 times the size of the common housefly, but it's gray. It's got 3 stripes on its thorax. And if you can see its abdomen, it's kind of got a checkerboard. But we're going to talk about these 3 mainly because they get in the house this time of year. They're the larger flies, so not the smaller ones like the fruit flies and fungus gnats. That's a different talk, but they all have this sponging mouth part. And these are the places where they often are found and where they breed. Not only are these nuisance pests, but because they have this sponging mouth part, they are actually able to transmit pathogens because they must regurgitate digestive juices on what they're landing on, which is usually unsanitary. And then they need to ingest liquid. So they break it down and they suck up what they're feeding on. And so that can mechanically transmit these pathogens that cause Salmonella or Shigella or E. coli. And they've been suspected, the flies, to be able to transmit diseases to humans like typhoid, dysentery, and cholera. So they are a public health pest. And if you ever have a fly in your house or around your windows, you'll know that they will leave regurgitation spots and fecal spots, which is pretty dirty and nasty. But we know, I guess, as an entomologist, we'll eat a lot of things that maybe bugs have been in, and sometimes we even eat bugs, but many entomologists will not eat things that flies land on. So I would take that as a hint that you shouldn't probably eat that. And we'll tell you a little bit why. So flies go through complete metamorphosis. So that means their immature stage look very different from their adult. The form, the function, what they're feeding on, where they live, where they hang out, and what they're doing. So the female fly will lay eggs in decaying organic material. Often it has to be very high in moisture so the eggs can develop and hatch. These larvae are often called maggots when it comes to flies. They'll molt a few times. They'll eat and eat and eat this unsanitary stuff, go into this pupal stage. It looks like a capsule. Sometimes people will find these little dry puparium. And over time, then they will emerge as winged adults. And you can see they're very different because this adult has wings and can fly, very mobile, can spread those germs around. So it's important for us to know this life cycle so we can break it, because an effective IPM program, we want that to be long-term. We want it to be preventative. And so knowing those life stages and where we can find these filth flies is just very important to focus on. And so their life cycle can be completed from egg to adult as little as 7 days to over 45 days. And that will be dependent on the quality of their food resource, which is usually pretty nasty stuff, which is everywhere, the type of fly, and the temperature or environment. And I've just got some pictures here of, dead rodents and maggots moving, pupa in mouse traps, and then adult flies. And a lot of times we'll see these flies as pollinators outside. And at that time, they're not being very pesty. Pests are organisms that you don't want where they are and often cause some problems. And that usually has to do with, the population and location. So that's why it's important to keep these outside where they belong and away from your home. So when it comes to IPM solutions, we are going to focus on prevention because that's something that we can do as homeowners. And then when they do get in, then we want to try mechanical control first. And if you do need some form of chemical control, I would recommend contacting a professional for like a certified and licensed person who knows how to use those products according to the label. But sanitation, it's just what it sounds. Cleaning up is a form of pest management, cleaning up food spills and residues on the dishes, reducing that moisture that may be around with leaky pipes. Pet care, include that to be our inside pets, like, cleaning our cat litter. And I don't know if you've got birds or hamsters or lizards, all of these things create litter and waste, and we want to make sure we manage that. Trash management, very important. If you've got compostables or recyclables, making sure that residue is cleaned out so flies aren't inclined to come in. Exclusion is a huge part of any pest management program. A lot of pests come through the doors just like we do. So we want to make sure those are closed, the windows are closed. And if we have things open, to have really good screens that will keep those filth flies out. You can conceal gaps where utility pipes go in or wires. And also when you're outside eating, because we all love, patio weather, to cover our food when we can, and, keep them off of our food. Habitat destruction is also very important. Most of these filth flies will start outdoors. So you want to pick up your pet waste from outside. If you've got like chickens or animals outside, manage the manure so that it's not going to be super wet, and the compost, manage that. And then also, removing the trash and decaying organic matter and dead animals. If you live near a street, you may get some roadkill, and also, things that just end up dying in the yard. You want to keep that disposed. When you get a fly in your house, the best thing to do is either get a shop vac and vacuum them up around the windows, or you can use a fly swatter. You just need to be very patient. They have very good sensory, with their big eyes, and they can sense that air movement. So you want to be able to sneak up on them and do it like with the wrist. If you need a tip, put on a light in like a bathroom and turn the rest of the lights off, and the fly will go in there, and then you can have a better chance of hitting it with a fly swatter. You can use fans so they won't land on your food, and in some cases you can use insect light traps. I know they have some commercial for houses, and then there are also outdoor traps that are non-toxic. They don't smell the greatest, but that's what attracts the flies to them. I'm supposed to give you an IPM action item for this long weekend, and that is to, grab a drink, a coffee or whatever, and walk around your home and do an inspection. Get to know your home, get to know the defects. If you have any, find out what needs to be fixed, screens, doors, seals, any types of defects, and then seal those up. Not only will it be good to exclude those filth flies, but it's going to be that time of year where you've got your home invaders or fall invaders or overwinterers coming in, like brown marmoset, stink bugs, box elder bugs, cluster flies, lady beetles, etc. As an entomologist, there are literally hundreds of thousands of described fly species. Not all of them are pests, so get out there and learn about them and figure out which ones you want to keep out of your house.
Awesome job, Jody. Thank you so much for that IPM minute. I think you might rate the highest on the yuck factor of all the presentations we've had. But you bring home the point that these are filth flies for a reason, and having the sanitation outdoors and indoors is important for mitigating their population. So thank you, Jody. I'm going to put one more link in the chat here for additional resources from the University of Nebraska at Lincoln. And at this point, we are going to end our session for today. So I want to thank both our speakers for their great presentations. We will be posting a video within the next week or so, and we hope that everybody has a terrific long weekend. So take care and thanks.